Which statistical test has saved the most lives? Well, I'll tell you about one that certainly saved millions later. But first, let's explore why we need to use statistical tests. Why we can't just look at summary information like averages and percentages and decide whether or not there is a pattern. When someone on a TV advert tells you that 8 out of 10 cat owners say their cats prefer Super Sammy snacks, you probably figure that they didn't ask every cat owner, especially if you're a cat owner and they didn't ask you. If you push to think about it, I'm sure you would assume that they only asked a small subset of owners about their cat's culinary preferences and base their answer of 8 out of 10 on that. The question, what proportion of cat owners say their cats prefer Super Sammy snacks, is being asked about all the cat owners, in Stats Talk, the population. But it's being answered using only a subset of the cat owners. In Stats Talk, a sample. The process of obtaining the sample from the population, that's known as sampling. Answering questions by sampling is generally the only practical approach, and a well-chosen sample will reflect what's in the population it's come from. But samples aren't usually exact mini versions of their populations. There is generally a difference between a sample and the population it's come from. This is called sample error. Let's take the question, how many jelly babies of each colour are there in this bowl? In this case, we could actually measure our entire population, that's all the jelly babies in the bowl, but let's just take a sample and see what that tells us. If we arrange our sample like this, we have a frequency distribution. To simplify things, let's just focus on one colour of jelly baby. In this sample, 6 out of 20 are orange. That's 30%. Is this the same as the population? Let's take another sample. In this second sample, the percentage of jelly babies that are orange is 36%. So now we know that the proportion of orange jelly babies in at least one of our samples is different from the population. Let's run that a few more times. Are any of the samples mini versions of the population? Let's make a frequency distribution of the whole population to find out. There we are, done. Turns out that 20% of our population was orange. Let's return to our samples. There, one was 20%, but look, it ranged from 7 to 36. There's a huge range. You might be saying, that's interesting, but so what? In 1950, Dole and Hill published a paper in the British Medical Journal which reported data on the smoking habits of cancer patients in the UK. One of the results they reported was that 97% of people with lung cancer smoked, but only 92% of people with other types of cancer smoked. The proportion of smokers amongst the lung cancer patients was higher. But was this a real pattern with something of biological interest going on, or just a product of sample error? Dole and Hill used simple chi-squared tests to show that it was very, very unlikely that the patterns in their data were caused by sample error alone, and that something of biological interest was going on. The rest, as they say, is history, and I'd say that puts the chi-squared test in the running for the title of the statistical test that has saved the most lives.